All right, ladies and gentlemen, welcome back. So today we're going to talk about some of the basic components, basic processes that happen during meiosis. That being said, just like we didn't spend copious amounts of time in the notes going over every detail of mitosis, we're going to do the same thing here. I'm not going to go through every detail of meiosis. I'm going to kind of give you the broad strokes that hopefully that is enough to trigger your memory from your underclassmen biology courses. We'll do some activities to kind of refresh your brain and go over some of the big pieces. But that being said, if things aren't coming back to you, please make sure that you see me and we can go through them individually. All right. Okay. So when we talk about meiosis, right, a couple of things to remember is that this is ultimately going to proceed as two rounds of cell division. Um, the only time that the chromosomes replicate is during the initial interphase. So we're still going to go through that cell cycle. Interphase will happen G1, S, G2. But when we get into that division phase of the cell cycle, the division phase is going to happen twice, but there really is no second interphase for any real intensive purpose. And so that division only happens during that initial interphase. And that's really important because the whole purpose to meiosis is to reduce the chromosome number. We're taking a diploid cell and creating a haploid one. And the whole purpose there is to ensure that the chromosome numbers aren't duplicating or doubling every generation. Cells generally don't do well with that. Some prokaryotes, some plant cells are a little more flexible in the amount of genetic information that they can handle and continue reproducing there on out. But especially when we look at animal cells, they don't do well with that. I've given you the example before of Down syndrome, right? A single extra chromosome worth of information, and we see quite a few developmental and physical differences from those who have a normal 46 set of chromosomes. So your cells have not evolved a way to manage that extra information. And so meiosis is there to keep this from happening. So when we look at those sets of cell division, it's going to occur as meiosis 1, which really is the reduction division. We're going to take that diploid cell, the homologous chromosomes are going to separate, and by the end of that first round of cell division, we have two haploid cells. And those haploid cells are genetically different from each other, and they are genetically different from the original parent, not only in the sense that they have half the chromosome numbers, but even the arrangement of those chromosomes is going to look different by the time we get through the end of meiosis 1. Meiosis 2 then takes that duplicated chromosome and now splits the sister chromatids and results in 4 haploid cells that are now genetically different on every level. They no longer have the replicated chromosomes either at this point. So when we look at meiosis 1, this is just a basic diagram representation of what happens. So you've got your sort of hallmark things that happen in prophase. The spindle fibers are forming, they're connecting to the chromosomes, the centrioles are moving to the poles, the nuclear membrane is disappearing. But the big hallmark here that makes prophase 1 different from both prophase 2 in meiosis as well as prophase in mitosis is here's where crossing over is going to happen. So here's where we're going to do our first step of mixing up, recombining genetic information between your maternal homologous chromosomes and the paternal ones. We get to metaphase 1, and now the spindle fibers are all attached to those kinetochores. Everything is lining up at the metaphase plate. The centrioles are at the poles, ready to pull these guys apart. Big difference here, though, is we're going to pull apart the homologous chromosomes. The sister chromatids are still going to stay attached here. And metaphase 1 is also where we're going to talk about independent assortment. So the way that those homologous chromosomes line up in terms of maternal information versus paternal information is completely independent from one homologous pair to the next. And so when we look at both the crossing over and the independent assortment that happen in prophase 1 and metaphase 1, this is why even if you have a full-blood sibling that is the same gender as you are, that they don't look completely identical to you, even if you've got the same set of parents, because we're mixing up genetic information and it is completely random as to what information ends up in which egg and which sperm. And then there's also randomization in terms of which egg and which sperm get fertilized to create that offspring. We get over to anaphase 1, and now again those sister chromatids are remaining attached, but those kinetochores are chomping down and moving those sister chromatid pairs, those homologous chromosomes, to opposite ends of the poles along those spindle fibers. We get to telophase 1 and cytokinesis. All right, and now cleavage furrow is going to form if we're talking about an animal cell. The nuclear membrane starts to come back, but we now are going to end meiosis 1 with 2 cells that are haploid. And that is going to quickly turn into meiosis 2. If there's any real interphase, if you even want to call it that, between meiosis 1 and meiosis 2, big thing is to remember that it's very short, very brief, and there is no genetic duplication. We're not duplicating chromosomes here. That would be completely counterproductive. We get to meiosis 2, and now I have got 2 cells going through that process, and the things that are going on in meiosis 2 look a lot more similar to what happened in mitosis. So prophase 1, centrioles moving to the poles, spindle fibers attaching, nuclear membrane disappearing. We get to metaphase 2, and now those chromosomes line up in the center, but remember each cell now only has 23 chromosomes. Anaphase 2, those sister chromatids now separate, move along those spindle fibers, and then telophase 2, we now reform those nuclear membranes, and those chromosomes relax back into chromatin, and now I have 4 haploid daughter cells that have formed, all of which are genetically unique compared to each other because of that crossing over and that independent assortment that happened back in meiosis 1. We can also look at some differences that occur depending on whether we're talking about sperm production or egg production. When we look at males, it really is sort of that single germ cell going through both rounds of division and then ultimately producing 4 sperm cells, what we would talk about as spermatogenesis. So here are your germ cells, right? They go through that round of genetic duplication. They go through meiosis 1 to produce 2 secondary spermatocytes, both haploid at this point, go through meiosis 2, and then they ultimately differentiate and mature into those sperm cells. What really looks different is the female version of this, the oogenesis, or the formation of that ova, or the egg. So while in the production of sperm, the cytokinesis is even. They take 1 parent cell, that germ cell, into 4 sperm. Females have to go through an unequal cytokinesis. And think about this. Dad's sperm really just drops off genetic information, and that's the end of his contribution to that offspring. The female egg, however, is ultimately going to become baby's first cell. So not only does mom have to contribute her half of the genetic information, but her egg is also going to contribute to that baby's first set of mitochondria and first set of ER and ribosomes and cytoplasm and all the molecules that go along with that. So when she goes through and produces those eggs, here's her initial germ cell. We're still going to duplicate genetic information, but when she goes through those 2 rounds of cell division to produce those immature ova, she ends up producing not just 4 cells, but instead 1 really functional egg cell and then these 3 polar bodies. Those polar bodies are really just genetic information, and there isn't much else left there. So those polar bodies actually get reabsorbed by the body, broken down, and they are not kept. And then here's another interesting thing. While males will produce sperm once they hit sexual maturity and can basically produce sperm until they die, a female will actually be born with all of the immature eggs her body will ever produce. Once she reaches sexual maturity, then her body is triggered to start going through this differentiation piece once a month, where her body will release a single egg for potential fertilization. Those polar bodies have already disappeared, they don't exist anymore. And she only has a finite amount of eggs that she can produce. So, yay for being female. We get to experience that menstruation once a month, and eventually our ability to produce offspring is stopped by menopause.